Welcome to section 4 of metabolism. In this section, we'll be discussing the tricarboxylic acid or TCA cycle. Sometimes you may also hear this called the Krebs cycle or the citric acid cycle. Let's get started. The TCA cycle is a metabolic pathway that converts acetyl-CoA to carbon dioxide. In the process, it generates NADH and FADH2, which can be used by the electron transport chain to produce ATP, which is cellular energy. GTP is also produced in the process. And finally, it's important to know that this pathway takes place in the mitochondria. Okay, with this in mind, let's look at the pathway. This figure is from section 1 of metabolism and provides an overview of the key metabolic pathways. In this section, we'll be discussing the TCA cycle, which can be seen right here. Okay, let's zoom up on the pathway. This is a detailed figure of the TCA cycle, which can be found in section 4 of metabolism. Notice that some of the reactions are reversible, as you can see by the bidirectional black arrows, while others are irreversible. The 3 colored arrows that are red represent the 3 irreversible reactions. Also notice that I've indicated what B vitamins are necessary for each of the reactions. You can see the small colored dots all over the map, like right here, for example, and right here. And these correspond to B vitamins shown in the legend right here at the bottom of the image. Okay, with this in mind, let's review the pathway. The first step is irreversible and combines acetyl-CoA with oxaloacetate to form citrate. This is done through the enzyme citrate synthase. Citrate synthase is inhibited by ATP, which you can see right here, and this should make sense. If the cell has enough energy, then it will block this enzyme so the TCA cycle doesn't continue to create more substrate used by the electron transport chain to make energy. It's also important to know that citrate regulates glycolysis and fatty acid synthesis. When the cell has a lot of ATP, the TCA cycle is inhibited and citrate levels rise, so it's a marker for excess energy. So citrate means there is sufficient energy. With this knowledge, you should be able to deduce how citrate regulates glycolysis and fatty acid synthesis without memorizing this information. It inhibits glycolysis by blocking the enzyme PFK1, which we discussed in the video on glycolysis. This makes sense. The cell has enough energy, so it stops glycolysis. So as you can see right here, citrate inhibits PFK1, which decreases glycolysis. Conversely, it activates fatty acid synthesis by activating the enzyme acetyl-CoA carboxylase. So I've shown that citrate increases acetyl-CoA carboxylase and thus increases fatty acid synthesis. This should also make sense to you. If there's enough energy in the cell, then the cell should store it in the form of fat. I talk about this more in the video on fatty acid metabolism, as well as the regulation of PFK1 in the video on glycolysis. In the second step, citrate is converted to isocitrate by aconitase. The third step converts isocitrate to alpha-ketoglutarate using the enzyme isocitrate dehydrogenase. This is one of the rate-limiting steps of the TCA cycle and therefore one of the most important steps you should know. Notice that isocitrate dehydrogenase is the first step in the pathway that produces NADH. It also produces CO2. It's important to know that this enzyme is inhibited by ATP and NADH. Again, you shouldn't have to memorize this information. ATP and NADH are both indicators of cellular energy, and it should make sense that these inhibit the TCA cycle. Likewise, it should make sense that when the cell needs energy, this enzyme should be activated. When ATP is depleted, ADP levels rise, so therefore ADP is an activator of this enzyme. I'll draw that in right here. Also notice that isocitrate dehydrogenase uses niacin or vitamin B3 as a cofactor. You can see that by the yellow dot right here. Notice niacin down here. Also notice that NAD right here uses vitamin B3 as a cofactor and that this is converted to NADH. The fourth step converts alpha-ketoglutarate to succinyl-CoA through the enzyme alpha-ketoglutarate dehydrogenase. Alpha-ketoglutarate dehydrogenase is actually very similar to pyruvate dehydrogenase, which is discussed in a separate video. And just like pyruvate dehydrogenase, alpha-ketoglutarate dehydrogenase requires several cofactors for optimal activity. These include thymine, lipoic acid, CoA, FAD, and NAD. These can be remembered with the mnemonic TLC for Nancy, tender loving care for Nancy. Notice the corresponding B vitamins with this mnemonic. T for thymine, L for lipoic acid, C for CoA, F for FAD, and then N for NAD. The B vitamins are incredibly high yield, and deficiencies of these vitamins are usually used as a way to test you on the TCA cycle. As you can see from the image, alpha-ketoglutarate dehydrogenase requires vitamin B1, vitamin B2, niacin, or vitamin B3, and vitamin B5. You can see them all right here underneath the enzyme. Notice that this step also produces NADH and CO2, and it's important to know that just like isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase is inhibited by NADH and ATP. Additionally, it's inhibited by succinyl-CoA. Similar to citrate, when the cell has a lot of ATP, the TCA cycle is inhibited and succinyl-CoA levels rise, so it's a marker for excess energy. It's also worthwhile knowing that alpha-ketoglutarate dehydrogenase is activated by calcium in skeletal muscle, which you can see right here. Recall that during skeletal muscle contraction, calcium levels rise, so it should make sense that during utilization of skeletal muscle, the TCA cycle would be active. This allows more energy to be produced for skeletal muscle use. Okay, let's do a question. A 47-year-old homeless male is brought to the emergency department after being found unconscious on the side of the road. He is resuscitated and complains of difficulty walking and remembering past events. On exam, he is ataxic and has ophthalmoplegia. What enzyme in the TCA cycle is likely altered as a result of this patient's condition? From the question stem, you should have noticed that this patient has Wernicke-Korsakoff syndrome. This is a syndrome that results in a number of symptoms, including memory loss, so remembering past events, ataxia, so difficulty walking, and ophthalmoplegia. Wernicke-Korsakoff syndrome is caused by thiamine or vitamin B1 deficiency. Let's pull up the TCA cycle pathway and see what reactions would be affected. Notice from the figure that alpha-ketoglutarate is the only enzyme in the TCA cycle that requires thiamine as a cofactor. So alpha-ketoglutarate is the most likely affected enzyme. In addition to Wernicke-Korsakoff syndrome, you should know that the other two conditions associated with thiamine deficiency, which are dry beriberi and wet beriberi. Dry beriberi can result in neuropathy and muscle wasting. Wet beriberi will have the same symptoms plus heart failure. Okay, let's continue discussing the pathway. The fifth step in the pathway converts succinyl-CoA to succinate, and this produces GTP in the process. The sixth step converts succinate to fumarate by the enzyme succinate dehydrogenase and produces FADH2 in the process. Recall that succinate dehydrogenase is complex 2 of the electron transport chain. Also notice that because FAD is a constituent of vitamin B2, this step requires vitamin B2. The seventh step converts fumarate to malate by the enzyme fumarase. The eighth and final step converts malate to oxaloacetate by the enzyme malate dehydrogenase and produces NADH in the process. Notice that this step also requires niacin, or vitamin B3, as a cofactor for the enzyme and in order to produce NAD. Okay, let's review with another question. A 24-year-old woman with a history of anorexia nervosa presents with fissures at the corners of her mouth and severe inflammation of her lips. On exam, she has corneal clouding due to vascularization. What two reactions in the TCA cycle will likely be altered as a result of this patient's condition? From the question stem, hopefully you noticed that the patient has inflammation of the lips and fissures at the corner of her mouth. This is called chylosis. Chylosis and corneal vascularization are key findings in patients with vitamin B2 deficiency. Anorexia nervosa can result in vitamin B2 deficiency. Let's pull up the TCA cycle pathway and see what reactions would be affected. From the figure, we can see that B2 is a necessary cofactor for alpha-ketoglutarate dehydrogenase, so a lack of vitamin B2 would result in inhibition of this reaction. Also notice from the figure that the conversion of succinate to fumarate requires vitamin B2 in the form of FAD. Without this vitamin, this reaction would also be inhibited.